In this video, we're going to review the characteristics of carbon as well as some of the details that we need to know for macromolecules. Understand that we're going to go back into some of the macromolecules in more detail in later units. For example, when we talk about enzymes, we'll go back and revisit proteins. When we go back and talk about DNA and RNA replication and transcription, we'll talk more again about the structure of nucleic acids. So this is just kind of an intro to both of those things. So the first thing is we have to make sure we understand carbon. And why is carbon such an important part of cells, living organisms, a lot of the molecules that are used in the processes that we talk about throughout the year? So carbon has an atomic number of 6. And if you remember from atomic structure, remember that means there are 6 protons and 6 neutrons here in the nucleus. That first energy level or valence shell can hold a max of 2 electrons, which means the outer valence shell or valence level, whichever terms you want to use, can hold the remaining 4. Now, if you remember, in order to be stable, an atom needs to have its outermost energy level or valence shell filled. And so carbon has 4, but it can hold 4 more. So it's halfway in between. It's not going to be strong enough to completely take away electrons from other atoms. It's not going to be, it's not going to want to give away its electrons either. So carbon instead tends to form a lot of covalent bonds. And remember, co means with, so covalent, there is a sharing of electrons with another atom. And so carbon does a lot of that. Carbon forms a lot of covalent bonds. Most frequently, we find carbon bonded to things like hydrogen or oxygen or nitrogen. That's not to say it can't bond to other things, it's just that tends to be what we most frequently see. Now, with atomic structure, we remember, or molecular structure, sorry, we tend to just look at chemical formulas. We tend to just draw them out very simply. But we need to also remember that we're talking about 3-dimensional molecules and compounds, and so it actually looks a lot different. So that's why sometimes you see structures that look like this or like this. Now, with all of those covalent bonds that carbon can form, it's important to understand that some of those covalent bonds can be single bonds, and when we see them in diagrams, it's just represented by a single straight line. Or they can be a double bond, meaning 2 atoms are shared between these 2 carbon atoms, for example, so we see it represented as 2 lines. Or it can be a triple bond, meaning they are sharing a total of 3 atoms between those two carbon atoms. Sorry, they're sharing a total of 3 electrons, not atoms. But anyway, so again, just make sure you understand a single line is a single bond, a double line is a double bond, and then 3 lines is a triple bond. And so carbon can form all of those different types of covalent bonds. Other characteristics that we need to understand about carbon is because it can form, all of these covalent bonds, it's a big part of, or the backbone of, a lot of these macromolecules. The macromolecules that we're going to talk about in this unit, which are the carbohydrates, the lipids, the proteins, and the nucleic acids. So for example, here is a structure showing DNA, and you don't see them here in the diagram just because it gets, one of the reasons is they write it or they draw up this structure is the carbon is there at each corner. So it's a 5-carbon chain or a 5-carbon ring, and then there are things bonded together or bonded off of it. And so you can see that it's the backbone or the basis for this DNA chain. Okay, same thing here. This is an amino acid. Carbon, again, is the backbone or the basis for this amino acid. And as this starts to connect to other things and form a longer and longer protein, or what we initially call an amino acid chain, again, you've got more and more carbons that are involved. And then here we've got an example of a lipid, and so the carbons would be in here in this in the black part of the diagram right here. All right, Other things that we need to know about carbon. So not only does carbon form up to 4 covalent bonds, it forms single, double, or triple covalent bonds. It's part of the backbone. Well, the shape can also vary as well with the structures that it makes because of being able to share, or I'm sorry, being able to form those 4 covalent bonds. So it can form chains. So all of these right here would be chain structures. It can form ring shapes, which are basically these circles, or it can be branched, so it can branch out and have different parts to it. Carbon can also form something called an isomer, and isomers are molecules that have the same molecular formula. So for example, we talk about molecular formulas like glucose is C6H12O6. That's a molecular formula. So these isomers can either be structural, meaning this has 1, 2, 3, 4, 5 carbons here. This also has 5 carbons. They have the same number of hydrogens, but structurally they're different. So if we were writing out the chemical formula, they would be the same, but the structure is actually different. So that's called a structural isomer. Another example would be sort of a spatial difference. And those are called cis-trans isomers, and it just has to do with what is attached to the carbons. So for example, you can see the different groups or the different things. X just represents what could potentially be attached to that. They would be attached on the same side, whereas in a trans isomer, you've got the potential things that are attached to the carbon backbone on opposite sides. So that's what a cis-trans isomer is. So one is the things are attached on the same side, the other one, the things are attached on the opposite side. And again, the X just represents all the different possible things that could attach to these potential molecules here. And then the other one are what are called enantiomers. I never know if I say that word right, but anyway, they are mirror images of each other. And so mirror images, for example, you see these hands here. So they look the same, but they're kind of opposite of each other. So they're mirror images. If you put them on top, you put the hands on top of each other, they're pretty much the same. Okay, that's what enantiomers are with their molecular structure. They are basically just where things are attached to the carbon are mirror opposites of each other. And then the other thing that we need to know when we just talk about organic molecules or carbon-based molecules, we need to know that there are things called functional groups. And functional groups are groups or chemical groups that are attached to these carbon chains or these carbon rings or carbon structures, and they give certain properties. Okay, so we have what are called hydroxyl groups. Hydroxyl groups, we usually see it written as an OH. However, depending on how it's attached, it could be written as an HO because it's the oxygen that's actually bonding to the carbon. We have a carbonyl group, which is a carbon double bonded to an O. So just C and O, but there's a double bond there. We have a carboxyl group, which is COOH, an amino group, which is NH2, a sulfhydryl group, which is SH, a phosphate group, which is oxygen, phosphorus, 3 oxygens. So basically, you're not necessarily going to need to recognize this part. It's this part that you're usually going to see. So just a phosphate with 4 oxygens surrounding it, and one of those oxygens is double bonded. And then a methyl group, which is CH3. So do you really have to memorize functional groups? No, you don't. But it's helpful to know and recognize them, and just know, again, that they're going to give certain characteristics. Okay. All right, so moving on to talking about macromolecules now. We need to remember that macromolecules are really big molecules that are made up of thousands or hundreds of thousands of smaller molecules. So I like this diagram here because it's the example I always use because, most people play with Legos. And so these individual pieces or these individual Legos are monomers. Okay? We're just talking about a general process, so we're not specifically talking about carbohydrates or proteins or anything else. So we have a bunch of these smaller compounds and they get put together. Okay, they get chemically bonded together and they start to form bigger structures. So once you put a bunch of monomers together, then it forms what's called a polymer. So mer, think of the word mer, or the root word mer means part. Mono means one, so there's one part. Poly means many, there's many parts. So here's a polymer that is made up of 3 monomers. Here's a polymer that's made up of 5 monomers. Can it be bigger than this? Definitely. This is just an example. Okay, and so we need to understand that a majority of macromolecules are polymers. They are made up of smaller things. Now, how are these macromolecules put together? There are 2 basic processes, really important. You definitely need to know these and understand these. And you need to remember that when you put the monomers together, because remember, everything is all chemically balanced, all the electrons are bonded, the atoms are happy, all the spaces are filled. So in order to put 2 monomers together, we've got to remove some stuff so that way there's room for a chemical bond. And so that's a process called dehydration synthesis. And so the things that you need to remember about dehydration synthesis is you're linking or putting together monomers. Synthesis means to make. So linking and synthesis, we're making something. But the dehydration, remember what happens when you're dehydrated? It's basically there's a loss of water. So what happens, and we can see in this very generic example here, you've got a hydrogen from one monomer, a hydroxide from another monomer. Those are being removed, and then what happens is you have these open ends now that can form bonds, and those are now attached. And you remove a molecule of water. And so that's why it's called dehydration, because we have formed and removed a molecule of water from these 2 monomers, and it's synthesis because we have made a polymer. Okay, so we've linked it together and made a polymer. So that's dehydration synthesis. So again, remember, synthesis means to make, and we're making something by linking it together, by removing a couple pieces so we can link it together. And the dehydration part means we're removing the atoms to make a molecule of water in order to form this other polymer. Now, in order to do the opposite of that, if we want to break down a polymer into smaller pieces into those monomers, then it's going to have to break that bond. And so again, using this generic diagram here, remember, we've got to make sure that the oxygen here, the carbon here, we have to make sure that their outermost energy levels are stable. And so what happens is it uses, and so most of these processes that we talk about are cells. So these cells are using water molecules to do this. Sometimes it uses the help of enzymes, sometimes it doesn't, depending on the process. And so it's going to break apart this water molecule into a hydrogen, which will attach here, and a hydroxide, which will attach here. And so now you have the oxygen nice and stable, the carbon here is nice and stable. And so that's a process called hydrolysis. So again, hydro refers to water. Lysis means to split or break apart. So we're using a molecule of water to split or break apart that polymer. All right, so now let's get into talking about the 4 categories of macromolecules. This chart is here because we need to remember that the major macromolecules are proteins, nucleic acids, lipids, and polysaccharides, or what we refer to also as carbohydrates. The monomers that are put together through dehydration synthesis to make these things are amino acids, make proteins, nucleotides make nucleic acids, fatty acids and glycerol make lipids, and then monosaccharides make polysaccharides. And again, there's that mono and poly. All right, so let's talk about carbohydrates first. Carbohydrates do a lot of different things inside the cell. One of the things that we need to remember, they are the most important energy source for almost all cells. Okay? We need to know what atoms or what chemical elements are found in carbohydrates. We need to remember it's basically different amounts of carbon, hydrogen, and oxygen, so CH and O. And the ratio that we usually see carbohydrates in is a 1 to 2 to 1 ratio, meaning for every 1 carbon, there's 2 hydrogens and 1 oxygen. So again, the most common example that people remember is glucose, which is C6H12O6. So it's a 1 to 2 to 1 ratio if we reduce that C6H12O6. Now, when we look at carbohydrates, they're generally going to be in one of two categories. You have sugars, which are used for immediate energy, and then you've got starch, which is used more for a storing of energy. And starch is going to be more commonly found in plant cells versus instead of animal cells. Now, when we talk about naming, so this fancy word right here, nomenclature. Nomenclature is referred to naming. So when you name something in science. Most sugars have an OSE ending. So for example, sucrose, we see this OSE ending. Lactose, OSE, that tells us it's a sugar. And so monosaccharides there's different types of monosaccharides. So glucose is a monosaccharide, galactose, fructose, and so they, again, all have different formulas that are ratios of CH2O. Okay, so again, here's the example of glucose. Oops. So disaccharides, prefix di means two. So saccharide, think saccharide is sugar. So disaccharides are two monosaccharides linked together by a covalent bond, again, going through that process of dehydration synthesis. And depending on the types of monosaccharides involved, they're going to depend on what type of disaccharide is produced. So for example, you have a glucose plus a glucose, that creates something else called a maltose, a different type of sugar. Another example, glucose and fructose make sucrose. So again, different chemical combinations of things. Polysaccharides are going to be much bigger, and so a lot of different monomers have been linked together, and starch is a really good example of a polysaccharide. And again, remember, it's pretty much going to be the source of energy storage for plants. Glycogen, on the other hand, that's going to be the carbohydrate storage in animal cells. Okay, so when we have glycogen forming in our bodies, for example, in our cells for long-term energy storage. Cellulose is another type of polysaccharide, which is, instead of energy storage, it's used for structural material. Whoops, that should be down here. Oh well. And then there's another type of polysaccharide, which is called chitin. I know it looks like it says chitin, but it's pronounced chitin. And that is the structural material that makes up the exoskeleton of a lot of arthropods. So things like, I'm totally drawing like spiders and things in shellfish that have that hard outer shell. That's because it's hard and it's protected, and that's chitin, which is a structural component or a structural polysaccharide. All right, so the storage polysaccharides, remember again, are used to store that extra glucose. Okay, remember starch is a polymer of glucose, so lots of glucose molecules have been bonded together. There are two different types of glucose, and one is called alpha, the other one is called beta, and it just depends on where the hydroxyl group above the first carbon. So with organic chemistry, they get really super fancy. And the carbon, and again, you don't see it drawn here. They don't write the C because it's implied. Okay, it's just the way these structures are written. So there's a carbon here, 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 and then there's an oxygen here. So everywhere there's a carbon, you see the blue numbers. So this would be carbon 1, this is carbon 2, carbon 3, carbon 4, 5, and 6. Now, the only difference between the alpha and beta glucose, if you look at the structure, is where is that hydroxyl group positioned. So here it's below the plane of the C1. In the beta, it's above. So that's what they mean by is it below or above, okay? And it is kind of confusing because you would think above would be, above and below would be below. No, it's the opposite. So again, I'm not quite sure how often we're going to see that, but just if you do, that's what it is. All right. Now, glycogen, again, glycogen is much more extensively branched out than starch to make it more efficient as far as storing energy, because remember, when we have all these branches, that means lots and lots of chemical bonds. And so it makes it more efficient. And as the body breaks down the glycogen, that energy from those chemical bonds is released and it can be turned into ATP. Now, in animals, including humans, glycogen is stored in the liver and in muscle cells. And so we store enough glycogen for less than a day of normal activity. So that's why, we have to eat to also provide energy. And so when blood glucose drops, the amount of glucose in our body, the glycogen that's stored in the liver and muscles gets converted back into glucose to help keep things going. Structural polysaccharides, so remember, there's storage and structural. So going back to structural, remember this is just an example of what I was talking about when I said that cellulose is a big part of structure for plant cells. Because remember, plants do not have internal skeletons like we do to grow up against the force of gravity, and so they're very dependent on that cell wall as well as that large internal vacuole. Now, roughly about 50% of all the carbon, the organic carbon in the biosphere, is tied up in cellulose. Okay, and so plants produce roughly about 100 billion tons. So 10 to the 11, that's 1 with 11 zeros. That's 100 billion. So plants produce 100 billion tons of cellulose per year. And a lot of the organic carbon that's produced, about half of it, is tied up in that cellulose. All right. Sorry, this is what I was talking about when I was talking about the chitin in the exoskeleton. So just some examples of different things. Now, one of the reasons why, some animals can eat certain types of plant matter and get energy versus animals can't eat the same has to do a lot with the enzymes that break down those different types of, alpha and beta glucose. So cellulose has a lot of beta glucose. And so a lot of the enzymes, for example, that we produce, our bodies can't break it down. So celery, for example, has a lot of cellulose. We eat it, but that's why only, there's that term roughage. So a lot of the food that we eat that has a lot of fiber or roughage, that's the cellulose base that our bodies can't break down, that just passes through our digestive system and comes out. Okay? But it is a really important part of our diet. All right, so that's it for carbohydrates. We're going to talk about lipids next. Lipids generally are referred to as fats and oils. Now, things that we need to know about lipids is lipids are not polymers, but they are still fairly large molecules. Okay, so they're not necessarily made up of large, or a bunch of smaller monomers. We need to remember that they are made from carbon and hydrogen. Because they're made from carbon and hydrogen, they are hydrophobic. Remember, phobic means fear of. So if you have a phobia of something, you have a fear of something. So that means they don't break down in water. Okay? And so examples of lipids are things like fats, oils, waxes, phospholipids, which we'll talk a lot about when we talk about cell membrane structure, and then also steroids. And so the main purpose of fats and oils is for energy storage, and they contain almost double the amount of energy as carbohydrates. Now, we need to remember that a lot of lipids are formed by combining a glycerol molecule with 3 fatty acids, and that's something referred to as a triglyceride. So here is a triglycerol molecule, here's a fatty acid, fatty acid, fatty acid. Okay, so when you see a structure like this, that's a triglyceride. And so the carbon atom is joined to another carbon by a single bond. So that's what's referred to as saturated, meaning there's no double bonds. There in those fatty acids, that's all single bonds. And so that's what we refer to as saturated. Okay? It means every single possible hydrogen that can be bonded to something is bonded to something. So fats are going to contain the maximum possible number of hydrogen atoms. So they're straight chains. They have no double bonds. They are produced mostly from animal products. They tend to be solid at room temperature. And so grease, lard, butter, these would be examples of fats right here. Okay. Whereas when we talk about oils, if there are any carbon-to-carbon double bonds in that fatty acid, so here's one double bond. Okay, what it does is it creates a kink or a bend in that chain. And so that is what we refer to as an unsaturated fat. Okay, it just means that it doesn't have the max number of hydrogen bonds that it could technically hold, because there's a double bond someplace else that's not allowing for the extra hydrogen. So fats mostly are produced by animals. Oils are produced mostly from plant products, whereas fats are generally solid at room temperature. Oils are generally liquid at room temperature because of those double bonds or those kinks prevent the close packing of those molecules. All right, so canola oil, corn oil, peanut oil, those are things like that. Now, as far as, generally it's like, well, wait, why is peanut butter, which comes from plants, why is it a solid? Well, peanut butter is a solid because when we produce peanut butter, generally it's got stuff added to it. So that's why if you read the labels, you see the term hydrogenated. Basically, it means they've added extra hydrogen atoms to it, and so they've helped to unkink those fatty acids, and so that's why peanut butter can be more of a solid. Even though it's spreadable, that's still technically a solid. All right, phospholipids. Remember, phospholipids make up the membranes in all organisms. They're very, very similar in shape to triglycerides, except for instead of having 3 fatty acids, one of those is replaced with a phosphate. And so a phospholipid, which we need to remember, is a glycerol, 2 fatty acids, and a phosphate group. And so these phospholipids have part of its structure that we refer to as sort of the head, which is hydrophilic. Remember, philic is loving, so hydrophilic, water loving, and it has to do with the negative charges in the phosphate. So because those negative charges are there, it can interact with water, not a problem. Whereas the tail part, since the tail part has all the hydrocarbon bonds, those are nonpolar, so they don't like water. They don't interact with water, so those are hydrophobic. So in a cell membrane, you see all the hydrophilic heads on the outside of the cell membrane, and you always see the hydrophobic tails on the inside. And remember, there's going to be water in the extracellular space. There's going to be water inside the cell, and so the parts that don't like water are going to be sticking together. All right, steroids. Steroids are, again, still technically lipids. Instead of having those straight chains, though, like the phospholipids and the triglycerides, they are rings, and they're ring structures. There's 4 interconnected carbon rings with a fatty acid chain that's attached, and so that makes them hydrophobic. So again, hydrophobic, cholesterol, they don't like water. Now, we tend to think of cholesterol as a bad thing because, we hear about having too much cholesterol in our diet, but cholesterol is an important part of our cell membrane. It's just the problem is the way that we eat and the foods that we eat, a lot of the processed foods, a lot of the meat, a lot of, the meat that we eat and everything else, like it's giving us too much cholesterol, which then starts to accumulate in our blood vessels and then start to harden and start to block off arteries, and that leads to a lot of heart disease. And then waxes are basically long chains of lipids that are connected to an alcohol or a carbon ring. And waxes are really important for a lot of plant structures, animals that have feathers, because that functions or that provides a lot of the waterproofing. When you talk about plant cuticles, which helps to provide from moisture loss through evaporation, and on feathers to help feathers from getting waterlogged, and so then birds can't fly. All right, so that's it for lipids. We're going to move into talking about proteins now. Now with proteins, we need to remember, and again, we're going to go back and talk about proteins again in other units, but we need to remember that proteins have multiple levels of structure. So there's the initial level, which is looking at the amino acids. There's a bunch of amino acids that form a long chain, which is called a polypeptide, poly meaning many. Peptide is the name of the bond that connects amino acids together. And then you have other chemical bonds that form that will cause this polypeptide chain to start curling up or folding or knotting up on itself. And so there's different types of structures. Now, we need to remember that proteins are the primary structural and functional component of a lot of cells. Proteins are like the workhorses of the cell. There's tons of things that they do. Okay, they're, of course, the most important proteins as far as cellular functioning are going to be enzymes, because they help to regulate metabolism. They act as catalysts by speeding up reactions, so reactions that would normally be too slow in cells. They help speed up those reactions to keep the cell alive, and they're also structural components in cells. So they do a lot of different things. We need to remember that proteins are polymers, so they are very large molecules that are made up of a bunch of smaller amino acid monomers. Just some reminders about the enzyme. Remember and again, we're going to go back into this, but just remember that enzymes are very specific, and it has to do with the folding of those proteins and the different types of chemical bonds. And so that's why certain enzymes can only work with certain substrates or certain reactives, because it has to do with their specific structure. Okay, but again, we're going to talk about that in another unit. So proteins have a lot of different uses, essential functions in, just the human body. So, and a lot of these also are found in a lot of other animals as well. So again, they're involved with support, storage, transport. So hemoglobin is the transport of iron and oxygen and carbon dioxide in the blood. Communication, because it's involved with hormones, which are basically signals that are sent throughout the body to different receptor organs. They're structural parts of receptors themselves. They're part of movement when we talk about muscle cells, so actin and myosin fibers. They're part of the immune response, and then they're also, of course, part of cellular reactions. Now, we need to remember that there are 20 different amino acids. Do you have to know the 20 amino acids? No. Okay. So there's 20 different amino acids, and there are, I don't even know how many proteins. There are thousands of different proteins, and it's how the 20 amino acids are linked together. Just like when we talk about nucleic acids, there's only four bases, but there's millions of different possibilities because of how things are paired together. So what we need to know is when we talk about amino acid structure, everything is always the same. There's a carbon in the middle, there's a carboxyl group, there's that functional group that we saw before, there is a hydrogen, and then there's an amino group. So these three things, or four if you include the carbon in the middle, these things are always the same in amino acid. What is different is what's called the R group. The R group is just, instead of saying X, they call it an R group. Okay, so the R group is what's going to be different. So for example, in glycine, it's just a single hydrogen bond there. However, in alanine, it is the carbon bonded to a carbon, which has 3 hydrogens attached. Valine has a much more complicated structure. Leucine has a more complicated structure. Isoleucine has another one. Proline, tryptophan has got some ring structures. So you can see everything else is the same, in all of these different amino acids except for the R group. So if there's 20 different amino acids, that means there's 20 different R groups, but the rest of the amino acid structure is the same. And so we categorize amino acids by the R group. And each type is going to be categorized based off of kind of the environments and how it behaves. And so it's important for the structure of the proteins. Okay, and so of course the shape of the protein is going to be important for its specific function or job. So when we categorize these amino acids, you have what the polar ones. So you have polar R groups. Remember, polar means they are hydrophilic. They're fine around water. They can be on the surface of cells. That means the R group is going to have oxygen, nitrogen, or sulfur. There's the nonpolar group. Sorry, I'm pointing in the wrong spot. Nonpolar, right here. There's the nonpolar group. The R groups contain only hydrogen and oxygen. And so they are hydrophobic. They don't like the extra positive and negative charge from water. And then there's the charged group, meaning the R group carries an extra charge. Okay, so here there's an extra positive charge, and these two there's a negative charge. All right, so proteins. Again, proteins are formed by linking together long chains. So just like we talked about with lipids, just like we talked about with carbohydrates, we have that process of dehydration synthesis that's connecting all these monomers. And so what we have, again, is a specific type of chemical bond is formed between all these little dots, which represent amino acids, and that's called a peptide bond. And so because we have so many peptide bonds, what they call it is a polypeptide chain. So again, poly meaning many, peptide meaning the type of bonds. So a chain because lots of polypeptide bonds happened or were made, and so it's all linked together. So the shape of that protein, now that we have that polypeptide chain, is going to happen because a polypeptide is going to spontaneously, meaning the cell doesn't really have to do anything, it's just going to spontaneously form into a shape that's been determined, and that shape is going to be determined by the order of the amino acid chain. Okay, and so it's reinforced by the interactions between the R groups of different amino acids. So you have chemical bonds that are forming. And so when you have this, you've got, you have covalent bonds forming, you have hydrogen bonds forming, you have, sometimes it's going to be chemical bonds between sulfurs, and those are called disulfide bonds. And so you have different things that are happening. Now, just like, you learned about with DNA structure or DNA replication in first year bio, when you have a single change in a nucleotide, that can cause a bigger change. Well, the same thing here. If you have a single change in this, what we call the primary structure or the first level of structure, that can completely change the shape of the protein, which means it's going to have a different function. It's no longer going to have the same job. And so that can cause deformation of a protein. For example, a single substitution of a specific amino acid in red blood cells results in sickle cell anemia, meaning the cell is misshapen and it can no longer function the correct way. So, depending on where the change is, it can have big consequences. Okay, so different types of structure. Remember, primary structure, I have my polypeptide chain. There's a carboxyl group on one end. There's a, oh man, I just forgot what it's called. There's another functional group on the other end, sorry. So that way everything is nice and chemically bonded. You have interactions happening between all of these amino acids, and it's going to form a second type of structure. So it's not going to stay this nice straight chain. It'll form either what's called an alpha helix because it coils up just like a telephone cord, or it forms what's called a beta pleated sheet. And a beta pleated sheet is like if you take a piece of paper and fold it up into an accordion. And again, these folds and these coils are because of bonds and reactions that are happening between different amino acids. So that's secondary structure. Tertiary structure now is our coils and our beta pleated sheets, or whatever the structure is now folding up and knotting up on itself again. Quaternary structure is now you have more than one of these chains that have gone through this process are involved. So here's one, this is this one right here, here's a different one, and another one. So in this example, there are 4 different amino acid chains that have all been knotted up together. Okay, so we need to remember primary structure, secondary structure, tertiary, and quaternary. All right, so this is just some more detail as far as how secondary structure results, okay, whether it's the alpha helix or the beta pleated sheet. You've got hydrogen bonds that are causing parts of the segment to either coil or fold, so it's the hydrogen bonds here, whereas the tertiary structure is going to happen more because of the R groups of the amino acid. Remember, we talked about some of them being polar, some of them were nonpolar, some of them are charged, and so that's going to create different bonds, okay, different structures. Disulfide bonds are going to be part of tertiary structure, more hydrogen bonds. Again, so just know that knotting is because of bonds. Okay, so here's just a diagram that goes over examples of where you would see or what types of bonds you would have. So you've got hydrophobic interactions, meaning they don't like water, so they try to start to buddy up together to avoid water. You've got the disulfide bridge because or disulfide bond because it's a bond between 2 sulfurs. You've got hydrogen bonds that are forming. You have ionic bonds that are forming. So just again, some different examples. All right, again, quaternary structure, 2 or more polypeptide chains are now linked together to form that completed protein. Okay, now sometimes the shape of proteins is way too complex and it needs some help. Okay, so it's not going to form completely by itself. It gets the help of something called a chaperone protein. And so the chaperone proteins hold that polypeptide in the correct shape, so that way the chemical bonds can happen in order for the correct shape to eventually form. All right, now what happens if we start to interrupt the chemical bonds? What happens to the structure of a protein? That is something called denaturation. So when a protein gets denatured, then you have the disruption of all of those bonds or a lot of those bonds that are holding the shape of that protein. And so we need to know the things that are going to disrupt protein structure are going to be changes in pH, because remember, a change in pH, you're either going to have an increase in hydrogen ions, which are positively charged, or an increase in hydroxide ions, which are negatively charged. So that positive and negative is going to interfere with chemical bonds. Salt can disrupt ionic interactions because, again, salt, it has a lot of ionic bonds. There's positive and negative charges involved. Increasing temperature, because remember, when you increase temperature, you're exciting electrons, making more energy in the electrons that are basically being held by those bonds. So if the electron becomes too excited, the bonds will break. If we change environment from polar to nonpolar, or disulfide bridges can be disrupted by chemicals or temperature. So a normal protein is all tightly packed and coiled up, but denaturation means it starts to unravel and unfold. So again, think of, a raw egg. The outside part, or what we call the egg white, is clear, and it's all clear and very liquidy because the proteins inside that are all very knotted up. Okay, and so when we heat up that egg and we cook that egg, then those proteins unravel and they take up more space, and so instead now we see like a solid white, and then also it becomes much more solid. okay, because those proteins have all unraveled. All right, and then the last part is looking at nucleic acids. So I'm not going into a ton of detail with nucleic acids here because we're talking about that in a lot of detail when we talk about not covalent bonds, I'm sorry, DNA and RNA synthesis. Sorry, I'm tired. DNA and RNA synthesis. So the things that we need to know right now is nucleic acids are polymers. They're giant molecules made up of a bunch of smaller molecules, which are a nucleotide. So a nucleotide is the monomer, the nucleic acid is the polymer. There's two types. There's either DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. And so a nucleotide is a base, a nitrogenous base, meaning nitrogen containing base. And so it's either going to be adenine, guanine, cytosine, or thymine. Or if we're talking about RNA, instead of thymine, it's uracil. And so they're going to be different depending on their chemical structure. Nucleotides have a 5-carbon sugar. That sugar is either going to be the sugar ribose, which is part of RNA, or it's going to be deoxyribose, which is part of DNA. And then it's got a phosphate group. So if we're talking about ATP, on the other hand, or ADP, then it's going to have multiple phosphate groups, either 2 or 3. But if we're talking about DNA or RNA, it's one phosphate group. Okay, so that is it for the really long lecture on review for carbon and macromolecules.